Hello everyone and welcome to the cell communication lecture. We're going to talk about how cells talk to each other and what happens to the cell inside as it receives that communication. When cells communicate with each other, they can either do it through direct contact, like you see in the picture here on the right. One cell is receiving what is known as the kiss of death, which will initiate an apoptosis or programmed cell death response inside of it. And then on the left here, we have actually yeast cells, and they are changing their shape in response to a signal they received, a signal that diffused on over from another cell. And in this case, it's for mating. In Saccharomyces cerevisiae, yeast cells can exist either as haploid, meaning they have one set of chromosomes, or diploid, they have two sets of chromosomes. And when they are haploid, they are also either of mating type alpha or mating type A. These are kind of like sexes in humans. And you have to be of opposite mating type in order to mate with one another so that the cells can fuse together and form a diploid. But if two cells of opposite mating type are next to each other, how are they going to be able to sense that in order to initiate mating? What they will do is they'll each secrete a molecule, what's known as either alpha factor, which will come from the mating type alpha cell, and A factor, which will come from the mating type A cell. And the opposite cell will have a receptor on the surface of the membrane that is the same shape of the opposite mating type communication signal. So the mating type A cell, for example, will have a receptor that will perfectly fit the shape of alpha factor coming from this cell. And vice versa, the mating type alpha cell will have a receptor that matches the shape of A factor coming from the mating type A cell. In this way, they will be able to sense the presence of each other nearby. And not only that, once they each receive the signal that was sent out, they will start to change shape. They will start to elongate towards each other. We call this schmooing. And they will continue this way until they actually make contact and fuse with one another. This will form a diploid yeast cell with both mating A and alpha information. So this mating was only made possible through the signaling between two different cells, and they both used chemicals that were secreted out to communicate with the other cell. But there are different types of signaling. On this slide, we have examples of direct contact signaling. And then on the next couple slides, we have indirect contact signaling. So on this slide, there's a couple of examples for direct contact. So on the right, we have cells that are actually connected together via cell junctions. And even though each of the cells that are connected together are surrounded by either a membrane or a membrane plus a cell wall, the two cells can still communicate with each other because they have specialized cell junctions that allow that.
Specifically, those cell junctions are called gap junctions in animal cells and plasmodesmata in plant cells. And again, these are cell junctions that allow cytoplasm and solutes between cells. A different type of direct contact is when two cells, which perhaps are normally moving or migrating around, make contact with each other through the use of surface membrane proteins. And so one would have a receptor and one would have a protein that would fit that receptor like a puzzle piece. And that interaction would then trigger an event inside of the receiving cell. So this is much like when cells of the immune system make contact with cells of the same organism to identify self cells so that they don't attack them or eat them, if you will. It's also the type of interaction that could happen when cells make contact to induce contact inhibition, so cells will not continue to grow and grow and grow and grow where there's no more space. So these are both examples on the slide of direct contact, and they would allow signaling or communication between cells. In the previous slide, only one of those examples would utilize what we call messenger molecules, and that would be the first example where two neighboring cells are sharing cytoplasm and molecules through those special cell junctions. But in the next example of types of cell communication, the indirect type, they're all going to be using messenger molecules. So I want to take a minute to kind of talk about some general characteristics of messenger molecules, or we could also call them ligands. Technically, a ligand is just a molecule that binds specifically, again, it has to be that very specific interaction, to another molecule, and usually it's another larger molecule. We could also call them first messengers. So messenger molecules, first messenger, ligand. Let's write down some characteristics. First, these first messengers or ligands, they can either be water soluble or lipid soluble. So that means they could be made out of amino acids or lipids. So an example of an amino acid based ligand would be insulin. So that's a polypeptide. Lipid based, those are some examples would be like vitamin D3, estrogen, testosterone. Those are all lipid based messenger molecules. Or it could even be a gas, something like nitric oxide. Ligands can travel long distances, as we're going to see in a second, or just short distances. It just depends on how far away the two communicating cells are. Some ligands can do both, and also ligands vary in terms of their affinity for various receptors. So when a ligand binds to its receptor, the strength of that interaction can vary. In some cases, it could be a very strong, solid interaction, maybe one that lasts a while, and in other cases, it could be weak. Okay, so now let's take a look at the indirect examples. For indirect communication, we definitely have to use messenger molecules because now the cells are not making contact with one another. And so on this slide, we're looking at examples of short distance communication. So the cells are pretty close to one another. But again, they're going to use chemicals that diffuse between the cells, or these messenger molecules. Now we have two examples that are being shown here. The one on the left, this is known as paracrine signaling, where you have a cell that is secreting out molecules that are going to be talking to a target cell. And a major example of such short distance signaling is when you have a cell sending out growth factor telling the target cells to reproduce or divide. Before I move on, I want to mention a term I used, target cell. A target cell will have the matching receptor for a specific ligand. And this is important because when a secreting cell releases some sort of a ligand like you see here, that ligand can go anywhere, really. But it won't be able to be received by just any cell. It has to be a cell that contains the matching receptor, or a target cell. On the right, we have another example. Again, this is short distance signaling, where again, there's a cell that will be secreting a ligand or messenger molecule, and then there is a target cell that should have the matching receptor. And this specific example is synaptic signaling, where a secreting cell, and in this case, it's going to be a neuron, will release a ligand or signaling molecule, and in this case, it's going to be a neurotransmitter, to a target cell. And this is, again, short distance, so these are very close together, but there is still a gap there, so that ligand or neurotransmitter will have to travel across to be received by the target cell. So what if a cell wants to communicate with a target cell that is very far away? So maybe a brain cell wants to communicate with some kidney cells. That's a pretty long distance in the body. And so in that case, we're going to have to use indirect communication, but long distance indirect communication. And so, for example, here would be our cell. It's not a neuron in this picture, but here would be our cell that is going to be secreting out a molecule. And the target cell is very far away. So the ligand, in this case, is going to have to travel along what I like to call the freeway in your body, some sort of vascular system. So in this case, we're looking at the molecule traveling in a blood vessel. So this type of long-distance signaling is known as endocrine signaling. And we call the signaling molecules that have to travel such long distances hormones. One benefit of this type of signaling is that you can really broadcast a message. So in this picture, it gives the impression that there is one initiating cell secreting a signal and one type of target cell. But in actuality, because these molecules are going to be traveling through the circulatory system, they're going to pass a bunch of other cells along the way. There may be other target cells that can also receive the same message. So it doesn't just have to be a kidney cell. It could be a heart muscle cell or a cardiac muscle cell. You could have a variety of target cells as long as they have that matching receptor to receive the signal. So this is a great way to broadcast your message in a multicellular organism. Now that we've covered the different types of signaling, the next section is going to be talking about the actual process of cell signaling. And this will be a quick intro to that or overview of that. So there's going to be 3 main steps to cell signaling. Number 1 will be reception, and that's going to involve the signaling molecular first messenger, a receptor on the target cell. After reception occurs, the next step would be signal transduction. This is when that message that is received is going to be converted into a different message inside of the cell. And that will usually happen in multiple steps. The last step of cell signaling is the response. So this is going to be the event that the initiating cell, the one who sent out the signal, this is going to be the desired response they want from the target cell. So we'll take a look at that in the next section.